This is the lab lecture video for the quantification of sodium via sodium 23 NMR lab. So this will go through the procedure and glassware and equipment used for this lab. So this lab is going to be the first experiment where we use the concentration unit mass percent instead of something like molarity or milligrams per milliliter or something. We're using mass percent. We're also going to be testing the amount of sodium in a food item of some kind. So in order to, using the nuclear magnetic resonance instrument, the theory and calculations video goes in more detail about how the nuclear magnetic resonance or NMR instrument works. But the way we're going to be, what we're going to be doing in lab is in part A, making standard solutions of known concentration of sodium in the unit mass percent. That'll be shared between you and your partner and another group. And then in part B, determining the density and testing the amount of sodium in a food item. So it'll probably be Pedialyte or chicken broth. So the point of this lab is we're going to figure out how much sodium is in your sample and then compare your results to the results on the label. The instrument only measures the isotope sodium-23. So again, in the theory and calculations video, we'll go through that. Lots of calculations in this lab, but let's talk through the procedure and how to do it. So part A, there needs to be 5 standards made for your results to work. Unfortunately, the NMR instrument is pretty slow. So instead of having every group make 5 standards, those 5 standards are going to be split between 2 groups. So you're going to have kind of a partner group situation going on where one group does 2 standards and one group does 3 standards, and you can fight over who gets to do the extra one. So there are going to be calculations that have to be performed before lab begins on how much sodium chloride it takes to make each standard solution. So on the data table, the standard solutions, the 5 that are shared amongst the groups, are going to be either 0.5 or 0.25% sodium, 0.75% sodium, 1% sodium, 2% sodium, or 3% sodium. We don't have pure sodium ion as an option. That's not a thing. Ions always, this is a cation, right? Sodium plus. So there has to be an anion to make the charge neutral. That's the law of electroneutrality. We can't just have a beaker of just sodium. So we're going to be using sodium chloride as where we get our sodium ion. Well, that means we're going to have to figure out how much sodium chloride it takes to get those specific percentage solutions. So those 5 standards, again, you and your partner, and then your partner group will divvy up who's going to do which one. Since we're doing these solutions as mass percents, we don't need to use volumetric flasks because volumetric flasks are about volume. Mass percent is grams of one thing divided by total grams times 100. All we care about is grams. So we're just going to use beakers as our containers when we're making our solutions. We're going to be using the balances a lot because we care about mass. Okay. So I would suggest a 150 milliliter beaker for each of your standards. So once you know the mass of sodium chloride that is necessary, you're going to take the beaker and set it directly on the balance, close all the doors, and tare the beaker. So the beaker says 0. Then you're going to take a scupula and the sodium chloride provided and add a mass of sodium chloride close to the calculated value for your standard. Then you will close the doors and accurately record the mass of sodium chloride. Well, that's cool. That's the mass of sodium chloride. But we also have to have the total mass in order to do a mass percent. So what you're going to do is after the beaker is on the balance with the sodium chloride in it, and you've written down your mass of sodium chloride, you're going to take a DI squirt bottle and you're just going to spray DI water directly into the beaker until the total mass is somewhere around 25 grams. Once it's around 25 grams, stop spraying, close the doors, and then record the accurate mass. So in the data table, you will be recording the mass of sodium chloride and the total mass. The total mass is going to be the mass with the sodium chloride and the water inside of it. Okay. You'll do this for your 2 or 3 standards, whatever it is, and your partner group will do it for their 2 or 3 standards or whatever they have. Once we've got our solutions made, we need to make sure that the sodium chloride and the water are well mixed. You're going to take a glass stir rod and just mix up the salt and water. You're going to do this for all your standards, but we don't want to contaminate them. So take your glass stir rod and either You're going to rinse it with DI water and then dry it with a paper towel before you use it with another solution. Don't take it directly from one into another because that'll just contaminate. So great, we've made our solutions. Now we need to get them ready to go into the NMR. So the NMR, we have to have samples that are placed inside of what are called NMR tubes. So these are just the containers that we have to have the samples in for the NMR to recognize them. These NMR tubes will have plastic lids of varying colors. I've got red, I've got blue, I've got white, and I've got yellow is what's with me. So these NMR tubes are very skinny. They have little markings where you can write on them, but I have a really hard time writing something that's legible on these NMR tubes. So you're going to have at least 2 standards, and you're going to need to be able to tell them apart once you put them in the NMR tubes. So my suggestion is to get 2 or 3 different colored lids, and like I would say, yellow lid is for my standard 0.25, and red lid is for my standard 0.75, and make a note of that in my lab notebook, because again, I have a hard time writing on these. Maybe you'll have better luck. So we've got to get our solutions transferred into these NMR tubes. The problem is, again, they're really, really small. You can't pour them in. It's too narrow. So we are going to use what are called pasture pipettes. These are the pipettes that are used in organic chemistry lab, because in organic chemistry, they can't use the plastic disposable pipettes because organic chemicals dissolve plastic. So they have to use glass. These are disposable. They're one use only. When we're finished with them, we're going to put them in the broken glass box. But they do not come with pipette bulbs at the end. So we have to use bulbs that are made for these pasture pipettes. So you're going to add the bulb to the top of the pasture pipette. And then you are going to, well, I guess before we do this, first measure 4 centimeters from the bottom of the NMR tube. Like, so starting at the bottom, measure 4 centimeters up and make a line on your NMR tube. That's how full we need to make the NMR tube of liquid. I'm guessing eyeballing 4 centimeters isn't something you can do if you're not terribly familiar with the metric system. So grab a ruler, measure 4 centimeters up, and mark it. Then you're going to take your pasture pipette, squish the bulb, and then release the bulb once it's in your standard. And then you're going to use the pasture pipette, which has a long skinny end, to add it to the NMR tube. So what I don't want you to do is don't place the pasture pipette directly on like the NMR tube to where it completely seals it. That's going to create a vacuum and be a problem. So instead, hold your pasture pipette up a little bit so there's a gap between the edge of the pasture pipette and the NMR tube, and then just use the pipette bulb to add your liquid. If it's not enough liquid up to the 4 centimeter line, then just repeat this until you have 4 centimeters of liquid. Once you do, put a lid on, okay? You have to be careful because, again, these are glass and they're relatively sturdy. These NMR tubes can be reused, but if you use too much of your strength, you will crack them, so be gentle. Just repeat this with a new pasture pipette for each solution, and you can use the same bulb. The bulb can be reused, okay? But the pasture pipette is a one use only, one chemical only. Put it in the broken glass box and repeat this until you have either 2 or 3 NMR tubes filled with standard. That takes care of part A. Sorry, I had to do a quick pause. I had a visitor in the lab, which I didn't think we all needed to see. So we're taking care of part A. So now we're ready for part B. You and your partner will be assigned either Pedialyte or chicken broth. Your partner group will have the other one. So between the 2 groups that shared standards, one group of 2 will have Pedialyte, one group of 2 will have chicken broth. Okay. So we need to do a couple things. One is to transfer your Pedialyte or chicken broth into an NMR tube. So to do that, you're just going to pour the Pedialyte or chicken broth into a beaker, get a fresh pasture pipette, a fresh NMR tube, measure 4 centimeters from the bottom on the NMR tube, and transfer the chicken broth or Pedialyte directly into the NMR tube. Okay. So you'll do that for your chicken broth or your Pedialyte. So then you'll have either 3 total NMR tubes, 2 standards and a food, or 4, 3 standards and a food. Okay. At this point, you and your partner can go put your name on the board because you will be ready to use the NMR. You're not done with the entire lab, but your NMR tubes are all ready. So there's only one NMR, and so the groups have to go back, and this is like your group of 2. Okay, so you and your partner made your 3 or 4 NMR tubes, and you'll test those. Your partner group made their 3 or 4 NMR tubes, and they will test those. And at some point before the lab report is due, you will just trade data. You will get the numbers that you need between them. You don't have to wait around for your partner group. If you're done first, great. Okay. You'll write your name on the board, and I will come get you in the order in which your name is written on the board to go use the instrument to record the data. I'm not going to go through how to use the instrument in this video because I will do that in class. And the instructions are in the lab video or lab notebook for part C, and most of it's working with the software. So it wouldn't make any sense anyway because you have to have like the computer in front of you to know what to click. Okay. So let's say you're not the first group to write your name on the board. So you're waiting for somebody else. Well, you have more to do in the lab anyway, and that is to determine the density of the Pedialyte or the chicken broth. So we're going to do that the old school way, which means we're going to take a 10 milliliter graduated cylinder, and we're going to use that to measure the volume, and we're also going to use that and the balance to measure the mass. So start with your 10 milliliter graduated cylinder completely dry, and go to the balance, close all the doors on the balance, click the tare button so the balance is at 0, and then place the graduated cylinder on the balance, close all the doors, and record the mass of the graduated cylinder. Go back to your station and fill the graduated cylinder up to the very, very top line, to the 10 line, with Pedialyte or chicken broth. You can use a disposable pipette to help with that to get it right on the line. That 10 line is the 10.00 line. So I'm telling you your volume has to be 10.00 milliliters of chicken broth or Pedialyte in this. Okay. Once you filled it to the 10.00 line, take it back to the balance, close the doors, tare the balance so the balance says 0, place this on it with the chicken broth or Pedialyte in it, and then record the mass after you close all the doors. So in your data table, you have mass of graduated cylinder plus sample, mass of graduated cylinder, those are coming from the balance. Mass of sample you're getting by subtraction, and then volume of sample, everyone's is going to be 10.00 because I'm telling you, make sure the volume is exactly on that 10 line, which is 10.00. Using the mass of the sample and the volume of the sample, you can measure, or you can calculate the density. You will also need to look at the label for the Pedialyte or the chicken broth, whichever one, to record. Well, first you have to write down the name, like is it Pedialyte or is it chicken broth? What did you do? You have to write down the serving size, which is on the label, like the nutritional facts label that you've probably looked at with your food before. The serving size it depends on the container, and if it will be in milliliters or maybe ounces. So record whatever it says the serving size is. It'll be a volume amount. And then somewhere in the nutritional label, it'll say how many milligrams, usually it's milligrams, of sodium are in a serving. So you're recording that as well. So those are in the data table. They're not calculations. They're coming straight from the label. Okay. The rest of the lab are calculations. Okay. So in terms of cleaning up, you're going to, well, everything is going to go down the drain because it's salt, water, and Pedialyte or chicken broth. So it's all going to go down the drain. The NMR tubes, when you're finished with measuring the NMR, you're going to take the lids off and you're just going to turn them upside down and shake them over the sink until you get the liquid out. It's going to take a little bit of shaking. Okay. These and the lids will have a special container for you to put them in. They're very challenging to wash. So the ones with the sodium chloride, you don't have to wash them. Just put them away. The one that had the chicken broth or Pedialyte, once you've shaken out the chicken broth or Pedialyte, squirt some water in there and then shake it out. What we're trying to do is get the food things out. We don't want these to mold while we're waiting to be able to wash them because we have a washer for them, but we can only do a few at a time. So it's a slow process to get through all the NMR tubes. So rinse out the NMR tube that had the food in it a couple of times with DI water before you put it in the container. Okay. Anything that had just salt or salt water, like your beakers with the standard in it, can just be washed like normal and then put away. Anything that had the Pedialyte or chicken broth also can be washed like normal, but by normal, I also want to make sure I stress like pretty well. We don't want the food stuff to be stuck in the glassware and for it to mold. So the graduated cylinder, any beakers that had the Pedialyte or chicken broth in it, wash them really well. It also gets, stinky. So it's not just looking at the mold, but it's also stinky glassware. And this glassware is not going away. We're going to continue to use the glassware. So just the pass-through pipettes, these are going to go in the broken glass box when you're finished with them. These are going to go in the drawer where you got them. You can reuse these. So NMR tubes have a special place to go. Pass-through pipettes go in the broken glass box. Disposable pipettes get thrown away. And then everything else gets washed and put back. So a lot of this lab is waiting for your turn with the NMR. That's okay. It'll be good times. Half the class will be doing this lab and half the class will be doing a different lab. So your lab table will all be working on this NMR lab at the same time. and the other lab table will be working on a different lab.